Hello, this is a basic little introduction to forensic science DNA, presented by me, Frankie Chavez III. We'll get right into it. There we go. One of the big principles that modern forensics DNA is based off of is short tandem repeats. Um, so first off, a little basic introduction stuff too. DNA can be found in almost all human cells and stuff, besides red blood cells. Um, and the main basis that DNA testing is based on is short tandem repeats, or STRs. STRs are found in the non coding region of our DNA, which makes about 98% of it. And these short tandem repeats are basically just repeats of the different nucleotide bases, so GCAT, just like in random orders and stuff too. And since there's so much of it, they make really good biomarkers for DNA testing. So the main type of DNA testing is STR profiling, and it has a lot of different applications in forensic science. It's not only just used in criminal investigations, but it's also used for paternity testing and some other uses, and civil uses and stuff. Um, so here in the United States, I believe mandated by the FBI, there is a minimum of 13 STR markers used, or STR markers used, that we need for like a match. Also, STR profiling tests can take about 2 to 3 business days. So it's a relatively pretty quick test and stuff too. It has a good turnaround rate for it as well. So, there is a good reason why we do use 13 STR markers. I couldn't find the statistic exactly. I've seen it before, but I didn't want to state it and kind of talk out of my butt. But basically, just that having um, 13 STR markers like match means it has like there's like a very low probability of misidentifying someone. And also that 13 STR marker provides a very high discrimination rate for identifying people. So it's a really good way to um, kind of like pick out a specific person from the population stuff too. Ooh, also another good little tidbit: STR profiling does not work on identical twins, because basically they have the same DNA, so it would just come up the same for both of them. So this brings up another good topic. So the crime scene is not perfect. It is not the lab. Finding samples out in the field can result in varying degrees of evidence quality, either due to temperature conditions, due to weather, such as temperature and moisture levels. High temperature and high moisture are great ways to degrade DNA a lot, but also maybe there just isn't that much DNA left on a sample. Maybe it was left on a Q-tip, or maybe there's like a few drops of blood on some cloth. Um, Before then, usually this meant that this type of DNA testing was very limited in its application. So you either needed enough DNA, or in this case and stuff, if it was degraded enough, you just couldn't do anything with it. I think an example is like the OJ trial, with just how degraded some of it was by being left in the car. But now we have a new method of PCR that makes it to where we can use smaller amounts or heavily degraded DNA sample stuff too for proper DNA forensics. Now, what is PCR? PCR stands for polymerase chain reaction, and it's this really groundbreaking method that lets us essentially amplify DNA samples. Think of it as using like a photocopier. It's pretty simple. So essentially, you would have the DNA you want to copy in the area you're like. You would have in the lab, of course, and stuff too. So you'd have your primers, you'd have other nucleotides, and you'd have in some type of solution. You would heat up the solution, and basically that would remove the hydrogen bonds between the strands of DNA. And then after that, you would let it kind of simmer. The primers would then bond to the separated DNA strands, and then after that, there's enough nucleotides for it to start replicating stuff too. But also, you have an enzyme in there called Taq polymerase. What that does is it builds a new strand of DNA. And then after this completes and stuff, you just repeat the process over and over and over again. So you can have billions of DNA copies that are then suitable for DNA testing. These are some of the more basic principles of like modern day DNA testing, and a lot of stuff that you will be seeing in the forensics and stuff. So yeah, there you go. Thank you for listening, and I hope you have a great day. Bye bye.